My name is Liz Yohe, and I serve as the Community Health Index Coordinator through University of Missouri, but I partner with U.S. Forest Service and Missouri Department of Conservation. So today I'll be going over some of the history of Community Health Index. And let me see something. There we go. A project that was started to assess the ecological integrity of ecosystems in the Midwest with a focus on fire-adapted natural communities. So these would include tallgrass prairies, oak savannas, woodlands, pine sites, and pine oak woodlands. The two main goals behind the creation of this survey were, first off, to give resource managers the tools to prioritize the best sites for restoration, and second, effectively perform long-term monitoring to determine if our current methods are efficient or if we need to change them. The project was originally started through Mike Leahy with Missouri Department of Conservation and has become a partnership between Mark Twain National Forest, U.S. Forest Service Eastern Region 9 Office, and NatureServe. Right. So we are looking at natural community management. To do natural community management within an adaptive management framework requires 4 steps. The first is defining and describing what natural communities actually are, conducting inventories and assessment of natural communities, monitoring natural communities as management occurs, and reassessing the effectiveness of management actions. So this is where adaptive management comes into play. So natural communities or natural ecological sites are defined as groups of native plants, animals, and physical environments least negatively impacted by modern humans. So typical natural communities will have intact soils and have not seen high soil disturbance. So the 6 steps to develop a community health index. Number 1 is select the ecosystem type to be assessed. We need to identify the key ecological attributes of the ecosystem type. Select a level of assessment. There are 3 levels. I'll go over those in just a moment. Identify the metrics. Identify scores and thresholds for the metrics, and this includes field validation, and provide scorecards and reports that facilitate interpretation of what is driving the scores. NatureServe defines 3 levels of ecological integrity monitoring. So level 1 is going to consist of remote sensing. This is utilizing software such as GIS, so ArcGIS or ArcPro, or other aerial footagery. Level 2 is a walkthrough survey. So this is where CHI would come into play. Level 3 utilizes detailed plot measurements. So examples of this would be quadrat monitoring or FQA analysis. So the CHI attempts to assess 4 broad metric groups, including landscape context and patch size, native plant species composition, vegetation structure, and negative impacts such as non-native invasive species and local disturbance on the sites. So you'll note here that native plant species composition and structure does account for the bulk of the CHI score. This is because the vegetative community influences most ecosystem functions and provides habitat for other taxonomic groups while being the primary vector of energy flow through an ecosystem. There are strong correlations between vegetation and soils, and the plants are obviously the most easily and practically measured variables of a natural community. Vegetation also integrates spatially and temporally variable natural and management-induced disturbances, and there's also some level of correlation between vegetation and insect diversity. So this is why we measure the vegetation. Okay, so I'm going to walk you through the protocol of one of our community health indexes. We will use the Pine Oak Woodland CHI. This has been developed along with Mark Twain National Forest. And these are mixed shortleaf pine and oak sites in the Missouri Ozarks, which were never glaciated. We created this protocol using expertise from Missouri and beyond. We consulted ecologists, botanists, foresters, and wildlife biologists. We field tested the protocol and modified the protocol based on their experience and knowledge, as well as what our field results are showing. So it's an always evolving process. So the first step is to delineate sampling units. We utilize ecological, soils, and management maps to delineate these units. They're typically 40 to 60 acres, and a 40-acre site typically takes under 2 hours to sample. Prism plots are designated within the unit. These align with Missouri Department of Conservation and Forest Service protocols. So we designate 1 prism plot per 10 acres, but no less than 4 plots per unit. 3 timed meanders are conducted per sampling unit to assess ground flora cover. The time limit on each transect is determined by the size of the unit, but it's typically less than 90 seconds per acre. During the meandering route to the prism plot, plant species are called out to a data recorder, and these are marked off a list. And once the time limit is reached, the process begins again, and the plant list restarts each time. We created the list of plant species recorded along the time meanders to include species that were relatively easy to identify vegetatively during the growing season and are longer lived throughout the growing season. So we avoided early year ephemeral species. The plants are also, they must indicate habitat quality, and there's typically 60 to 80 species on a list. So I am assuming people are familiar with the coefficients of conservatism. Another term for this is C-value, but if not, here's just a really short explanation. Values are assigned per state by expert botanists to plants, and these values range from 0 to 10, depending on their level of conservatism. 0 to 3 species would be kind of the more weedy species. 4 to 6 are matrix species, so these can be thought of as the backbone of a site. Typically, you want to see a high level of matrix species, if not all of them that are on the list. And those 7 to 10 are going to be more conservative species that typically require high integrity sites to grow. Another example of this can be relic species. We selected both matrix species. So here are some examples. Matrix species would be like Andropogon gerardii, big blue stem, Symphyotrichum anomalum, blue aster, as well as more conservative species like Baptisia bracteata, which is cream wild indigo, and Eryngium yuccifolium, which is rattlesnake master. And typically the plant lists have about 75% matrix species to 25% conservative species. So initially, in the beginning of creating these surveys, we set sampling time periods at 5 minutes. But as you can see from this graph, after about 15 minutes, the number of new species found rapidly declined. For a 40 to 60 acre sample unit, 3 timed meanders overall capture most of the ground flora target species within a unit. So at each of the prism plots, trees are determined to be in or out using a 10 basal area factor prism. This does also include snags because they are very important for focal bird species in this landscape. Most people are familiar with the brown-headed nuthatch, which prefers dead pine snags for nesting. Canopy closure is also determined, and a qualitative assessment of vegetation structure and abundance of indicator species is recorded. In 2022, to better capture fire effects, we also began monitoring litter and duff abundance at the prism plots, and I'll go into that in just a moment. So overall, we look at woody overstory, understory, and ground layers. And this looks at abundance of grasses, forbs, and ferns at each prism plot. So a little bit about litter and duff abundance. As stated, this was not part of the original protocol, but was later added because this is an often overlooked component of natural community health. So I apologize for the bad picture. It's kind of hard to find a picture that better depicts this, but it gets at what we're trying to explain. So litter is going to be things, I don't know if you can see my mouse, but litter is going to be things like parts of leaves, but they're still going to be recognizable. So you can still recognize them as leaves, right? Or they may be parts of flowers that you can still recognize as flowers. Duff is going to be below the litter, and these are partly decomposed organic matter that you can no longer tell. We know that they're pieces of leaves, but we can't tell what kind of leaves or that they were leaves at all. So they're unrecognizable plant forms. And most of the time, an ideal prescribed fire should remove this litter layer while retaining the duff. While the presence of heavy litter can impede ground flora and seedling regeneration, duff is rarely a concern because it's quite thin typically and won't cause the same concerns as heavy litter abundance. But lack of duff and total litter removal to expose bare mineral soil can lead to soil displacement, runoff, and erosion, as well as having devastating effects on bacteria, fungi, and mycorrhizal fungi as well. In our survey, litter is measured to the nearest half inch, and duff is just assessed as being present or absent. Okay, so here are some examples of different understory and midstory growth levels and how they can influence the ground flora composition. So as you can see, there's not going to be, in this picture on the left, there is not going to be much of a ground flora layer because there's just not much sunlight getting down to that ground flora. And it's going to be the same thing for the picture on that upper right. On the lower right, we have a high ground flora composition because we're allowing sunlight to get down to that level. But the big takeaway here is that woody understory layers can dramatically influence herbaceous ground cover. So the final step of the CHI is going to be scoring. Each of the metrics gets a score. Landscape context is 10% of the score and is determined via GIS or aerial imagery. So as you can hear some examples, things that will drive your score up and down. You ideally want an area of surrounding landscape and native vegetation to be fairly high. And for the size of the woodland community, you're looking for basically the larger, the better. It's going to increase that score. Okay, so quick breakdown of the ground flora portion of the CHI survey. The number of matrix species are counted. So this is up to 46 matrix species. Those are going to be your 4 to 6 level species. So that's worth a possible 14 points. The evenness of matrix plant species, this is going to be how many times a species shows up in those 3 transects. If it shows up all 3 times, then you get a portion of those points for up to 4 points possible. The relative abundance is just looking at overall cover. So if you see a really, really high abundance of matrix plant species, that would drive up your points as well. And it's the same thing for the conservative species. So that'll be your 7 to 10 scoring species. And this does account for 55% of the total score. So keep that in mind. That's a pretty high portion of the overall CHI score. Functional group cover and woody structure accounts for 35% of the score, and that's broken down into 7 metrics. So these look at overall cover of the plant groups. This is including graminoids. Examples of these are grasses and sedges, as well as forb cover. Understory, so these would be your sapling species, or I'm sorry, your seedling species, and mid-story, which are going to be your sapling species, is also determined. So you'll note here, so for instance, this number 3% cover of woody understory that is less than 4.5 feet tall. In your first class, you're going to increase your points, but as you get to those higher classes, you can potentially decrease your points. So this is -6 here. And the same thing for mid-story. You start out with increasing your points and then you decrease your points. So the idea here is that you don't want a monoculture of any one component. You really need to have small amounts of all of these components within this site. And it's the same thing for rubus and rust species. These are going to be your blackberry and your sumac. So as you can see, it starts out with 3 points. So there is an acceptable amount of these species in a system, but when a system becomes overrun by blackberry or sumac, it will result in a negative. So that's loss of points. A set of forestry metrics is also assessed. This is going to be canopy cover, percent stocking, pine and oak basal area, the presence of snags, and whether there is pine and oak regeneration in the unit. So this is another big driver of scores at 11 points possible. And litter and duff presence is also assessed. So for the fourth component, we look at disturbance factors, and these are going to result in negative points. And this can take away almost 1/3 of the score. So these would include aggressive non-native species and any other factors that are degrading the site. For instance, here you can see evidence of feral hogs damage, because in Missouri, feral hog damage is a big concern, especially to these intact sites. Another big one is human-caused soil disturbance. So those are going to be things that are going to take away points. But the total CHI score will max out at 100 points. Total possible CHI score. There is a fifth portion looking at animal presence, and this is not included in that 100 points. So in theory, if you had all of these, you could have up to 110. So we look at bird and herptiles observed at the end of the survey. But obviously, these are going to require more time and sampling demands. However, if, for instance, you're walking through the woods and you run and you're doing the survey and you run into a timber rattlesnake, you can mark it down because it is going to show a higher quality site. If there's any kind of more intensive monitoring completed within the last 5 years, so these would be things like herpetile surveys, bird point data, things of that nature, this data can be used to complete this portion of the survey. So some examples of what would be included on the pine oak survey would be pine warblers, my favorite, timber rattlesnake, and indigo buntings. Oops, sorry guys. Yeah. So the field data is gathered and entered into an Excel spreadsheet with embedded formulas. This is nice because it can automatically calculate things like percent stocking, basal area of different species, things of that nature that are going to be reoccurring within multiple surveys. So this is what we use to calculate all of those results for us. Currently, we're working on a Survey 123 app that will fully automate the data entry and analysis in the field, as well as serve for data management. Basically, when you upload your information into the survey, it gets automatically loaded onto ArcGIS Online, and you can use that to handle all the data. So this is going to be really nice when we're able to use this full time. And managers can see which components of the CHI are driving their scores up or down, and this can tell them where to focus management. So once field data is collected and CHI scores are determined, how do we analyze this data to make management decisions? This graph is pretty simple, but it shows how sites with higher ecological integrity are ranked. So sites that have higher ecological integrity are going to be those A or B sites, while sites with decreasing integrity or experiencing degradation are going to receive C and D ranks. And ecological integrity overall is very important because these sites are often more resilient if they experience detrimental events such as invasion by non-native species or even high severity fire. Because these natural communities were already intact, they may be less negatively affected by these sites or may be able to bounce back faster. So sites that already have high ecological integrity are very important to protect. Okay, so we're going to dive into a little bit of our pine oak data. Between 2020 and 2022, we collected data on 59 pine oak sites throughout the Missouri Ozarks. Sites scores ranged from 26 to 66. The sites were broken down into percentiles and then further categorized in quintiles. So these would be 0 to 20, 20 to 40, etc. In previous reports, we categorized scores as poor, fair, good, but the quintiles are an effort to get away from that kind of subjective wording. Overall, sites that ranged in this 0 to 20th percentile experienced low graminoid and forb cover. They had higher mid-story cover and saw a smaller number of matrix and conservative species. So this makes sense. Those lower scoring sites just didn't have those components that these systems require to have that high ecological integrity. However, sites in this 80th to 100th percentile showed higher levels of both graminoid and forb cover. They showed significantly lower mid-story cover. So as you can see here in the 0 to 20th percentiles, we had an average of 51% mid-story cover within a unit. And by this 80th to 100th percentile, we had about 14%, so less than 20% cover. And then the matrix species and conservative species. So these are going to be your matrix species, and these are the conservative species, significantly increased from these lower scoring sites. So they almost doubled in the higher scoring sites. So makes sense. It's just nice to see that validation. Another interesting component is the average number of burns within the last 20 years. So in the lower scoring sites, we saw about on average, 2 burns per year. In the higher scoring sites, we saw closer to 10 burns within the last 20 years. However, those higher scoring sites already were given preferential treatment because they were higher scoring. So while it's kind of interesting to see this side-by-side comparison, it is important to keep in mind that the more you burn a site does not necessarily mean that you're going to have higher ecological integrity. There is kind of teeter-totter. You know, the more that you burn it, at some point, it's going to be great for the site, but after a while, you're going to overburn it. So for the briefest statistics lesson of your life, for anyone who's not familiar with the correlation analysis, the higher a correlation result is to 1, the better the relationship between 2 variables. So one of the questions that has been asked by people when they look at CHI is, are you collecting enough data within those timed meanders to even come close to what someone would find within when conducting quadrat level data? So we were curious about this as well. So we compared our data with quadrat data that was done on the same sites. So these quadrats are significantly smaller than our CHI units, but we made sure our CHI units laid within the footprint of where this quadrat data was conducted. And our results showed an R of 0.7 with a p-value significantly less than 0.001. So this means that our results are strongly correlated and statistically significant. So yay. But what this indicates is that a good amount of what is being found on quadrat level work is being picked up during a rapid assessment. However, it is important to keep in mind that rapid assessments may be more feasible given time and resources, but quadrat level data will always be more accurate. Rapid assessments will never replace quadrat level assessments. They're simply on two different levels of monitoring. So some tentative conclusions, what we've found so far with community health indices. CHI is a useful tool for teaching the components of what makes a healthy natural community. It is a coarse scale, but it is efficient at monitoring these sites. The CHI can be easily used by resource managers with some training, and it can be a great starting point for managers new to an area. CHI is not a substitute for detailed plot level work and statistical design to answer cause-effect questions or evaluate fine-scale changes in communities. What it can show you is trends, and that's all we're looking for at this point. And of course, further sampling is needed to assess trends in CHI scores with paired botanical plot scores. So the next steps for CHI work. In 2022, CHI was picked up by Forest Service Region 9 to utilize for their Southern Tier Oak Restoration Initiative. So this is looking at oak regeneration within a variety of different landscapes. You can see on the map here that this encompasses Mark Twain National Forest, Shawnee National Forest, Hoosier, Wayne, and Monongahela National Forests. And this project aims to provide targeted and effective oak ecosystem restoration within these sites. And CHI is being utilized to fulfill ecological integrity and monitoring within the story landscape. So this was a big step for utilizing these surveys. They're now being utilized on a broader landscape level scale, and it's pretty exciting to be able to compare all of these results across a much broader landscape than we were previously doing. And we're right now developing protocols for Shawnee, Hoosier, Wayne, all those other ones. All right, so some photo credits. And with that, does anyone have any questions?